to thank the organizers of the symposium to visit, to be in a place which, I mean, I was just beginning my career when Konowski left us. So I actually knew the name Konowski since a very, very long time. I had actually a promoter who was a very big fan of Konowski. So the name, both of the Nencki Institute and Konowski have been with me since my training at the PhD level. And so I'm extremely happy and honored to be able to speak in this room at the very moment that it becomes the Konowski Hall. And I've always been fascinated by this idea, the Gnostic unit, and the sort of joke that Ledwin made of it, the grandmother cells. And indeed, I did start my PhD. I actually started my PhD proving that there were simple cells in area 18 of the gut, as Bogdan may remember very well. But then I moved up the scale, and I went back from the back of the brain to the front of the brain, from the cat to the monkey to the human, and I'm now working in Parma mainly with humans and with mirror neurons and premotors. What I want to review here is work that I did when I was in Leuven, of which I am now an emeritus, which means in Belgian terms, please don't come back to your lab. And I had the good fortune to be able to build a very integrative lab, a little bit on the model of what happened here, where I could combine single cell recordings in the awake monkey, fMRI in the awake monkey, and then fMRI in humans. And this allowed me to describe a family of neurons, which are the gradient selective neurons, which are the topic of my lecture, which are on the way to the Gnostic or the very high level conceptual neurons. So they're somewhat further down. And I think one of the interesting lessons is that, just like Konowski, I'm extremely concerned that my work in the animal is applicable to you. And I actually spend time to develop techniques to really show that what I find in the monkey is actually directly relevant to humans. So that's more or less the story. So one, I very much remember when the first paper came out of the previous speaker, and actually had been one of the reviewers, I think. And from the monkey sort of perspective, that was very intriguing, to say the least. And so the usual interpretation was, but this is further along the system, as the previous speaker insisted, this is hippocampus, not infratemporal cortex. So it's a step beyond, and therefore you have, of course, multimodal, and IT would be purely visual. But I think what I want to offer today is that this is maybe only part of the explanation, that we should also think about species differences. I mean, it is the case that the human brain is far larger than any other, and certainly of the monkey. In terms of cortical surface, it's 10 times larger. If you would go to the mice or mouse, it would be 1,000 times. But even monkey is 10 times larger. So you simply have many more neurons. So you could have, I think, differences in sparseness of representation. So what we saw in the previous talk is a very, very sparse representation. And maybe you have to have many, many, many neurons to be able to follow that strategy. And that if you have a smaller brain like the monkey, you can have sparse, but not as sparse. But I think what I want to say is that even if it is not very sparse, still these are very sophisticated units. So we understand now relatively well the properties of these gradient selective cells, which basically encode the shape in the third dimension, which is a relatively high level property, which therefore fits this association idea of Konowski. Right. So just let me explain to you what I mean by depth order. So you're on the left, the eye, and you're looking at, you're fixating a point in space. Zero order depth is just distance, how far away it is from the fixation point. And that's interesting information, and mostly when people talk about 3D, that's what they think about, is distance. However, you can take derivatives. If you take the first order derivative, you get tilt in depth, which is somewhat ambiguous, just like orientation in 2D. It can mean that an object is inclined in depth, or it can be part of the description of the boundary in the third dimension of the object. And then you come, if you take the second derivative, you have curvature there, and that's always representative of the shape of the object in the third dimension. So we'll be talking about neurons that can encode these first order and second order gradients. That are the gradient selective neurons. Now we have several depth views. The most performant one is disparity, based on the difference in the position of the two eyes. And this is the only one that allows you to recover the sign of the curvature. So you can distinguish convex from concave. The monocular cues, such as speed differences, can only tell you that it is curved. It cannot tell you whether it's concave or convex. Still can tell you the local shape. And then we have texture and shading. And in fact, it will have turned out that we have, for the three first cues, we actually have gradient selective neurons. Just to make it a little bit, we'll begin with the speed cue. And so these are the stimuli that we have used in the last experiment. So the top left is a linear gradient, so it's an inclination where the left is in front and the right is in the back, or the opposite. Then you have a cylinder, which is a vertical cylinder where the middle is in front of you. And we could test that, of course, in different orientation. This is a bump where the curvatures in the two directions match. So this, you can see this as a convex or concave bump. And then you have a saddle where the two orthogonal curvatures are opposite. So these are the elements of description of the local 3D shape. All right. So the story begins some time back. The very first description we got of gradient selective cells was for linear gradients in speed, which we actually discovered in an area of the monkey which is called MT or V5, depending where you are in the world. And so we know since a long time that these cells are direction selective and speed selective. If now you take the optimal speed and the optimal direction and then you make a pattern of speeds, then you see that if you have a gradient where fast speeds are on the right, the length of the arrow gives you the speed here, and slow speeds are on the left, and the neuron is very happy. If you use the opposite gradient, the cell is not active at all, and you have a tuning for disparity. So this was the first description of gradient selective cells. What we could show is that this actually depended on the presence of an antagonistic surround. MT neurons have a complicated receptive field where the center is driving the neuron, and surrounding that center is what is called an antagonistic surround, which has the same often preferred direction and often the same preferred speed, but opposes the response. So if you make a stimulus that is larger and larger, you actually, very much like end-stop cells, you get a decrease of response for very large stimuli. And usually this is simply seen as a way to prevent the neuron to respond to large stimuli. So for example, if you would make and that's part of the use of it, is if you want to select a target to pursue it, then an antagonistic surround would be very useful. That was the traditional view. In fact, we found out that if you map in detail that surround, as shown here on the right, you actually find that the surround is not surrounding, as the name says, it's in a certain sense a misnomer, that often you have, it's reduced to a single suppressive region on one side. And of course, once you have such a structure, that's a perfect way to take a first order derivative. So we actually showed that these are all the selective neurons that we had for the speed gradient. They all had gradient, they all had the surround. If we masked the surround, then the selectivity disappeared, and there was actually a relationship between the direction in which these the suppressive regions were located and the preferred gradient. And we later showed that often these suppressive regions had a different speed preference than the center. So these are all the elements you need to build a selective neuron. One of the things that was missing in the original study was to show that this was invariant for mean speed, which is one of the things that we systematically tested in a study that was performed 10 years later, in which we could compare MT and FST, FST being the next level of processing because MT projects directly to FST. And so here you have an example of an FST neuron that is responding to mean speed, so this is homogeneous speed, not very much. All the first order tilted speed gradients, very little response. But you see that when you are using the second order stimuli, the 7 are here, so here we have the bump, the 4 cylinders and the 2 saddles. Then you see that these 2 orientations of the cylinder, the oblique and the vertical, are actually driving the neuron very well. There is some response to the saddles. These are higher order stimuli, more complex shapes, and they don't drive the neuron. You see here the response as a function of the different stimuli plotted for the 2 speeds. And you see that indeed there is a relatively good invariance for mean speed. So this really shows that it's the pattern of speed that is producing the response and not a value of the local speed. And so this is an example of selectivity for second order gradient. So this population of neurons allows you to analyze the depth profile based on the speed. This is to show that these FST neurons are not only selective or invariant, their selectivity is not only invariant for the mean speed as is shown here, but also for different positions in the receptive field. So here you have the map of the receptive field of the neuron, and here is the position of where the stimuli were placed in these 2 tests, and you see that the responses are very similar for these 2 positions. So this is a relatively high level representation that is invariant for the mean speed and the position just as you would like. This is a systematic comparison between a series of neurons in the same monkeys in MT, which is the lower order area, and FST, and you see that in general selectivity is present in both, but is more enhanced in FST. The selectivity index is here, so 0 would be no selectivity, this would be very high. The dark shaded areas are a significant effect of the different stimuli, so you see that the median is shifted to, this is for FST, that bar down here is MT, and you see that there is a shift to higher selectivity values in FST for first order and second order. You also see this is an index of the position invariance. Again, 0 is no position invariance here. At the right is high position invariance, this is speed invariance, and you see again these 2 indices are similar but always significantly higher in FST than in MT, which means that the neurons are more selective and more invariant in FST than in MT. But you have already a first representation in MT, which is then further elaborated in FST. I think that, so that's the first cue, the speed cue. I give you a few examples of the other cue, which is the disparity cue. So you can build the same representation of the shape in the 3rd dimension of the first order and second order gradients from a different cue, the disparity cue, which is the one that allows you to recover the sign of the curve. Here is behavioral evidence that actually monkeys are able to distinguish using disparity the difference between a convex and a concave shape, and if you use anti-correlation, that ability disappears. So monkeys are actually can use this cue. So here we have an example of a neuron recorded in the infratemporal cortex, in fact in a very small region that we call TES, which is in the lower bank of the superior temporal sulcus. And typically these neurons, if you show binocular stimulus, you have a selectivity for one sign of the curvature, in this case concave, and not at all to the convex. And if you show monocular stimulus, you don't have any effect, so these are clearly disparity selective neurons. The important proof that these are high order neurons that look at the gradient is to show that the selectivity for concave over convex is invariant over the position in depth, over the mean disparity. It's the equivalent of manipulating the mean speed. So this shows that again it's the pattern of disparity, not a given value of disparity that is driving the neuron. So this is a position invariant, but position invariant in depth of the representation of the sign of the curvature. So these neurons are in the infratemporal cortex, in the lower bank of the STS. You also find them in AIP, which is now a very high level area, not in the ventral stream, but in the dorsal stream. It's actually the most anterior part of the dorsal stream that is involved in grasping, as we know. And you find exactly the same type of neurons. Interestingly, this was predicted from something that you will see later which were our fMRI responses. We actually used the fMRI to guide our single cell recordings, because otherwise you can spend a long time finding out where sophisticated neurons are localized. So this is sort of the counterpart. If you look with very sophisticated stimuli, it could be that you're in the wrong area. You can spend the whole year looking for neurons, and this is sometimes has happened in the past. I think with fMRI in the monkey, we have now the perfect tool to guide the single cell recordings, and the phase patches, the work of Tsao and Freiwald are actually a beautiful example of that. Incidentally, Doris learned her trade in my lab. All right. This allows us to compare the surface, the 3D gradient representation at the end of the ventral system in TE, S, that small area in the infratemporal cortex, and in AIP. So we have the endpoints of the dorsal and the ventral stream. And then a number of interesting differences emerge. If you do an MTS, which basically is an analysis where you measure what the distance is between the different stimuli in neuronal terms, you find that in AIP there is a very gradual representation of the curvature of the, not of the sign, but the magnitude of the curvature. So these are increasingly curved stimuli, which are represented for both signs very systematically. In TE, you find something very different. There you have basically a representation of the sign, whether it's convex or concave, and not much of the magnitude. So you have a qualitative versus a metric representation in the ventral and dorsal stream. The dorsal units, this is the latency distribution of the AIP cells, which is at least 30 milliseconds faster than the infratemporal. So you have a much faster representation in the dorsal stream, because that is what has to guide actions. And then the representation in the dorsal stream is coarser than in the infratemporal cortex. For most of the AIP cells, it doesn't matter whether the curvature is really smooth or whether this forms an angle, whether you have hinged planes. So they don't distinguish this detailed variation in the curvature. There's hardly any difference. In infratemporal cortex, half of the neurons make that difference. So you have a much more precise and detailed representation in the infratemporal cortex. In AIP, it's coarser. All right. And so this just illustrates that we have a complete family of neurons, which I reviewed in annual reviews a few years back. So we have gradient selective cells for three cues: motion, stereo, which I showed. I didn't show texture, but if somebody is interested, papers again are published. And so you have both first order and second order selectivity, and you see that you have both dorsal and ventral stream. One group that has been very interested in this topic is the group of Sakata in Japan, and there are those who have studied the CIP neurons here in the first order stereo and texture. And then there is also the work in several laboratories showing that MSTD can also have speed gradient selective neurons. So how do we extend this to humans? We just recently looked at a number of patients and found that this is heavily disturbed in PCA patients, but perhaps this is not so clear. So 15 years ago, I developed in my lab in Leuven the fMRI in the wake monkey, together with Roger Toutel, Wim van Duffel, and Paul van Eck, a physicist. So basically, we had no money and only a very willing radiologist who wanted to share his horizontal 1.5 Tesla. So we converted the monkey chair in what is now known as the Sphinx position, which is shown here. So the monkey is attached here and is sort of sitting on his back legs and is facing the screen directly, unlike what humans do is usually a mirror. Here the monkey is looking directly at the screen. You can reward the monkey, he can respond. You can measure the eye movements, so they're fixating very carefully. And the other change we made is that we use a contrast agent, the Mayon, and here you see a comparison of activation for motion compared to static random dots with the bolt and with the Mayon in the same monkey and in the same sections, and you see that the activation is much better circumscribed, much more specific for the contrast agent, which also increases the signal to noise dramatically. So especially if you only have 1.5, this was tremendously useful. All right. And so the monkey fMRI is really the missing link. I mean, many people know about single cells, many people know about human fMRI, and they don't know how dangerous it is to go directly from one to the other. Classical assumption is, for example, the STS of the human exactly equal to STS of the monkey, which of course is absolutely not true. So you read something in between, and that is what monkey fMRI is. And that allows you to first go from monkey single cells to monkey fMRI, then you understand what the difference is introduced by the two different techniques. Once you understand that, then you can go to the human fMRI, then you keep the same technique, it's hemodynamic responses that you're measuring, but now you can understand what the difference in species are. We have actually made a lot of progress in understanding the homology between the cortex of the monkey and the human by doing these parallel imaging studies. For today, where we want to know where specific types of single neurons are located in the human brain, you need something in addition. And this is what I call the two-step strategy. So what you have to do is to develop an MR paradigm that is of course based on the cells of interest. So here the gradient selective cells. And you have to show that that paradigm drives specifically or activates the cortical area in which you have recorded your neuron. So it's sort of showing that you have a regional specialization. And then once you have proven that these particular areas, or one optimal thing is that you only have one area, is driven by that paradigm, then you move into the human, and where you find your activation has then a very good chance of being the homologous areas containing similar types of neurons. You know, that is very hard to directly demonstrate the presence of selective neurons with the fMRI. People have tried with adaptation techniques, which we have proven are not very faithful, and they also are multivoxel classification tools which are used very extensively, which may or may not work. People don't understand why they work. Until we have tested them in the monkey, it would be very difficult to understand what they really mean. So this is a completely different way based on the availability of monkey fMRI, which I strongly recommend to people who are interested in integrative neuroscience. Right, so how do we find this paradigm? So I will show you two examples, a paradigm for the disparity gradient cells and one for the speed gradient selective cells. So the first paradigm is for the disparity gradient selective cells, which we developed once we had found the initial neurons in the frontotemporal cortex. So you have a factorial design. So you have one factor is whether you show stimuli in depth, in stereo, or monocular. So you simply show the two left eye images, and then in the next presentation the two right eye images, they look flat. The other factor is whether in your 3D representation you have curved surfaces or flat surfaces. So here we show curved surfaces, and here we show flat surfaces at different distances from the fixation point. Here you see the profiles in depth, the 6 different profiles that we use in this stimuli here, curved stereo stimuli, which are shown at 2 different positions in depth. So this is sort of bird's eye view. So we have a wide variety of curved surfaces in depth. Then to equate as much as possible the stimuli, we actually calculate what are all the disparities that are present in our curved stimuli. And you see the black line is that distribution. And then we sample this distribution with constant disparity stimuli, which are then the ones we use here in these 3 here position in depth. So we can claim that both the range and the density of the different disparities that are in these 2 conditions are matched, and also that the number of different, because fMRI is sensitive to how many different items you show in a block. So here we have 12 different stimuli, we also have 12 different stimuli in this condition, 12 different positions in depth. And what we're after is the interaction between these 2 factors, which would mean that the difference between the curved and the flat surfaces is present for the stereo and not for the monocular condition. That's the region we are after, because that would correspond to the disparity selective neuron. And if you do this in the monkey, you find indeed that you have an activation very close to TS, where we recorded these neurons, as I showed you. You also find an activation in the AIP, where I showed you that there is also a population of disparity gradient selective neurons, and there actually is a 3rd region here in premotor cortex, in F5A, and we actually went there, guided by the fMRI, And believe it or not, you also have gradient selective, disparity gradient selective neurons in that area. So you actually have a very good match between the activations in that paradigm and the presence of gradient selective neurons. So that's the type of paradigm that we need. There's a small exception here in the back. There was a small activation in V3, which I think is a lower order activation. We could actually eliminate if we replace the control not with a single plane in depth, but with a step in disparity. So we think this is a lower order representation. But the three higher order all contain this disparity gradient selective neurons. If now you go to the human with exactly the same paradigm, this is the activation you find. So this is a typical SPM figure where you see the lateral brain of the left and the right hemisphere, which is called the rendered brain. So activations are projected on the surface, and you see this huge parietal activation, you see one temporal activation, you see two premotor activation, and then one in dorsal occipital cortex. So we actually know from many other experiments that AIP in the human is sitting here. And so this activation here, which you see here, of which you see the profile here, you actually here see the flat map. It's exactly the same data but presented on a flat map. A flat map for me is a far better representation because it integrates the whole hemisphere. Let me show, this is the calcari that has been cut open. So the occipital cortex, the back is here. This is the parietal occipital sulcus, this is the central sulcus, so this is parietal cortex, the IPS is actually here. This is the STS, so this is temporal cortex. So here is parietal cortex, and we know that this is AIP. So actually, and the profile is here. This region of activation corresponds exactly to the AIP activation that we have in the monkey, which was generated by the presence of these disparity gradient selective cells. So this gives you a strong indication, although we have never recorded directly, of course, in the human brain, you have a strong indication that similar neurons must be present here. We think that this might be the equivalent of the human equivalent of F5A, where you also have these neurons. And then this actually is too posterior to be the TES, or the equivalent of the TES side, which might be masked by the technical limitation that we have in the temporal cortex. So this might be a region that exists also in the monkey and that have escaped us in the monkey fMRI. Here you have a big region here of activation in regions that are in the vicinity of V3A. We actually now know that there are 18 retinotopic representations in the human cortex. And you have here maximum probability maps of these 18 areas on a flat map and overlaid on the myelin density. So this is work we are doing with David Van Essen in St. Louis. So you have here V1, you have V2, you have V3, and then you have a strip of areas here, V3A, V3B, V3C, and V3D. All of these correspond to that strong dorsal occipital activation. They have this interaction between stereo and curved surfaces. So we don't know, because it is very likely that some of these areas actually do not exist in the monkey. We think this is the equivalent of CIP. So V3 is here, in the monkey CIP and V3A are neighbors. Then all these areas are new areas. So here we don't know, because the monkey model sort of stops whether these are already contain higher level gradient selective neurons or whether it's still relative disparity. So this we have to be open, is still unknown. So here you have LO1 and LO2, which were introduced by Eager. Then here you have human V4, you have VO1, here you have the PITs, and then here you have the MT complex. You have MT here, this is now human. So we know where MT is finally in the human. You have putative MST, putative FST, and putative V4T. So you have this cluster. And this is now where we look for the speed gradient selective neurons. Remember, in monkeys they were in MT and FST. So here the paradigm is much simpler, and it's the one that I have been using since the beginning to try to reveal human regions engaged in structure from 3D structure from motion. This is simply a set of 9 random lines which are drawn randomly, and either, unfortunately, I didn't find the movie back, but either they translate at uniform speed, then they create an impression of lines that are more or less flat, or they rotate around the vertical axis, so they rotate in depth, and that creates a very vivid impression of 3D. So by subtracting the rotating minus the translation, you should get something that actually drives neurons that are involved in 3D shape motion. Now you remember, these are very different from the stimuli that I used in FST. So we actually record, so in the monkey, if we compare rotating random lines to translating, that's the activation you find. And the whole science paper was about the fact that we didn't find anything in parietal cortex, while in the human we did. But we also saw activation on the edge of the stimulus in V2, V3, and possibly V4. That was exactly like in the human, but we think that is an edge effect which doesn't really represent the stimulus itself. So the stimulus itself only was represented here in MT and FST. And we actually verified here, when we were recording from disparity gradient selective cells, speed gradient selective neurons in FST, we actually verified that they respond more to the rotating, which is the full line, than to the translating random lines to validate disparity. So here we actually know that it corresponds to the activity of the gradient selective neurons. And so now we port this to the human. Alright, so here you have another example of a gradient selective cell in MT. We have here the flat mat of the monkey now, and in fact there is exactly the same cluster. So these four areas form a cluster which share a central representation. So you have MT, which is here in brown, then next to it you have MSTV, then you have FST, and you have this very small region here which is V14. And so exactly the same cluster you find in the human as I have shown to you. And so if now you have an activation by rotate in the MR, which is on the right here, I'm running out of, okay, so this is the activation of, thank you. This is the activation in fixation condition by the rotating random lines in the monkey MT, and this is for the translation condition. The zero here actually is always the empty fixation. So these are really visual responses. And this is the same, but under conditions in which attention was manipulated, because the monkey had actually to detect the change in orientation of the fixation bar, like we used to use in the old single cell experiment. So you see that it's invariant for this attention manipulation. So if you now do exactly the same paradigm in humans, and you take the signals of MTV5 in humans, you get exactly the same difference. So from that similarity and the fact that these areas seem really to be homological, you can then conclude that in the human you have cells like this also in the human cortex. Of course, it's not a direct recording, which is something we are actually working on. All right, so this is a very quick and brief tour, but I'm happy to discuss, and actually everything is absolutely published. So what I have tried to tell you is that gradient selective neurons are a large family of neurons that encode 3D shape, using disparity, speed, or texture. We haven't found gradient cells for luminance, which would do the shading, and shading is probably a different mechanism. Importantly, they're located both in the dorsal and in the ventral stream. So, because of course 3D shape can be used both to recognize an object and to guide prehension. Using fMRI, you can devise strategies to infer the presence of this family of neurons in different cortical areas. So I've shown you examples of human AIP, what is likely to be the human homologue of F5A. I've shown you human MT, and probably similar neurons are also present in putative FST that we also have in the human. And I think it shows that the primate model is really directly applicable to the human brain. And it's not surprising because there's only a scale factor of 10, which is, as I said, very different from other species. And I think these sort of data show that indeed this animal model is valid for translational neuroscience at the neuronal and the areal level, not molecular level, but the neuronal and areal level. That's where the functions are. Thank you for your attention.